Well, hello, my name is Lauren Cervantes, and I am a third-year medical student at the University of Central Florida College of Medicine here in Orlando, Florida. And today we'll be discussing the differentially expressed genes in polycystic ovarian syndrome. And I will also want to thank my colleagues and the University of Central Florida College of Medicine for helping support this work. Hi, and myself or any of the other authors have no disclosures or conflicts of interest to resolve. So, polycystic ovary syndrome, also PCOS, is among one of the most prevalent endocrine disorders affecting reproductive age females with a global prevalence of 6 to 10%. It is characterized by hyperandrogenism, ovulatory dysfunction, and polycystic ovaries, and it's associated with an imbalance in hormones such as GnRH, LH, FSH, testosterone, and prolactin, mostly at the level of the granulosa cells. And so the pathogenesis of PCOS is complex, it is multifactorial, and it includes genetic, environmental, and intrauterine influences. However, very little is known about the genetic mechanisms of this syndrome. And so the objective of this project was to identify the differentially expressed genes in granulosa cells of patients with PCOS and hopefully offer insight into the pathophysiological understanding of the syndrome. So for our methods, we use search tag analyze research for NCBI's gene expression omnibus, also known as StarGeo. And we used it to identify the samples. It is essentially a tool that allows the reutilization of millions of samples available from previous studies in the gene omnibus repertoire. Within the software, a free text search was used to identify human granulosa cells, samples from patients with PCOS, and healthy control patients. A meta-analysis was then done via StarGeo to identify which differentially expressed genes within these samples were present in the PCOS samples as compared to the healthy controls. And then a further analysis was conducted through the Ingenuity Pathway Analysis IPA tool, with a restricted to a statistically significant genes of a p-value of less than 0.05 and an experimental log ratio of greater than 0.1. So essentially, StarGeo was the database search engine for us of publicly available genetic samples, while IPA mapped those genes to known biological pathways for us. Okay, and so for our results, we were given 23 granulosa cell samples from PCOS patients, and then 12 samples from healthy patients. And we chose to use granulosa cells because they're the supporting cells in the ovary involved in estrogen production, oocyte development, and they contribute to the follicular environment and are involved in cell-to-cell communication. So from these samples, 22,684 genes are identified. However, with our parameters, just under 200 met the criteria and were included in the IPA analysis. So in this table, we were given the top 10 upregulated and top 10 downregulated genes in these PCOS cell samples. Upregulating meaning that these 10 samples on the left, these 10 genes on the left are more expressed, are more active in PCOS patients, as compared to the cells on the right side of the chart, the downregulated, these genes are less active, less expressed in these PCOS samples as compared to healthy controls. So this diagram looks really complicated. However, it shows our top upstream regulator, which was predicted to be the lipopolysaccharide, LPS, which is a bacterial component known to trigger inflammation. So the orange note at the top, LPS, And all the arrows leading downward represent the predicted immune influence on the genes we found to be altered in PCOS granulosa cells. And on the bottom in the green and the red are genes that were in our previous table, which showing us the green ones being the more expressed and the red ones being the less downregulated in our PCOS samples. As for our orange lines are our predicted activation and blue indicating inhibition. And then our dashed lines being indirect relationships and solid lines being direct ones. So what we're seeing is that LPS activates a whole chain of inflammatory reactions. Things like IL-6, TNF, NF-kappa beta, all central players in immune activation. And the result is this pro-inflammatory signaling cascade inside the ovary. Even though there isn't an infection, these immune pathways are turned on as if they were, disrupting the normal hormonal and follicular environment of the ovary. Our activated Z-score was 3.642, indicating that this pathway is strongly predicted to be active and suggesting that the ovary in PCOS behaves like an inflamed tissue. The overall takeaway of this is that even though without bacteria, the ovary is acting as if it's fighting an infection with this inflammatory response. Here we have the second figure, which visualizes our top network, being the AKT signaling network, one of the most central pathways in our data set. In the middle, you can see AKT, a major regulator of cell growth, metabolism, and survival. All the nodes connected to it, mostly in green, are genes from our table that were upregulated in our PCOS granulosa cells. And each orange arrow showing us the activation and the blue showing us the inhibition. And this is important in many ways because it shows the cell-to-cell signaling interactions, which is how the granulosa cells are communicating. Hematological and immune responses connecting inflammation with hormone signaling, and inflammatory pathways that maintain that chronic immune response that we saw in figure 1. So if figure 1 was the fire alarm being pulled, figure 2 is the sprinklers in the building, even though there's no real fire happening. And so together, these findings suggest that inflammation and growth signaling are tightly linked in PCOS, driving abnormal follicular development and preventing normal ovulation. So for discussion, here's those top 10 upregulated and downregulated genes that we previously discussed. And so a literature review was conducted to research what current data is out there about all of these genes, what is their known functions, and how could they make sense in the pathophysiology of PCOS. And in doing that, we were able to make 2 groups to group these genes in. The genes that were involved in inflammation and immune dysregulation, and the genes involved in dysregulated follicular growth. And so now we're going to dive into each of these groups and talk a little bit about those genes. So the etiology of PCOS has been largely linked to chronic local ovarian inflammation, and which disrupts ovarian ovulation, triggers systemic inflammation, and contributes to what we know as hyperandrogenism and insulin resistance. So for our first bullet point, the gene CD163 has been known to play a crucial role in acute phase reactions and oxidative damage. It's possible that protective function may be sustaining the local inflammation and exacerbating the ovarian dysfunction that we see in PCOS. As for moving to the second point, B64 is a gene that plays an immune modulatory role in inflammatory conditions such as autoimmune arthritis, uveitis, and hepatitis, which may suggest that its function is contributing to the dysregulated macrophage-mediated immune tolerance. Our third bullet point, SF3R and LIN, are both genes that play key roles in neutrophil maturation, which they may be linking the immune hyperactivation to metabolic dysfunction and insulin resistance. Finally, GBP1 is a known upstream regulator of macrophage cell death and inflammatory signaling, both of which are contributing or may be contributing to the persistent inflammatory state that we see in PCOS. Now, moving down to the downregulated genes in the same group, CARMA1 is a key regulator in actin filament formation, influencing extracellular signaling, related kinase ERK, and p53 signaling. So its downregulation in PCOS may be contributing to that uncontrolled follicular proliferation, disrupting cytoskeleton dynamics that we need and are essential for ovarian function. And together, this group and these genes highlight how the ovaries' immune cells are staying active and they're interrupting the normal ovulation. As for follicular growth, dysregulated follicular growth group, we know follicular growth disrupts the ovarian homeostasis and contributes to the anovulation. And these differentially expressed genes include TGF-beta1, which is involved in extracellular matrix formation and is known to be highly expressed in granulosa cells. And so this upregulation may be contributing to matrix remodeling, ovarian fibrosis, and suggest a role in the pathogenesis of these polycystic ovaries. Second, SIGLEC10 is a regulator of the cell cycle. It has been associated with cell signaling and regulation, and it could be implicating the follicular development and ovulation, and can potentially serve as a biomarker for PCOS. And for downregulated genes, our first being CD24 plays a key role in ovulation by influencing granulosa cell signaling. Its decreased expression may be contributing to the anovulation and infertility that we see in PCOS. Second, MEM200C is a transmembrane protein. It's involved in reproductive functions, facilitates cell-to-cell extracellular communication, and its downregulation suggests to us that there are disruptions in the cellular signaling pathways essential for follicular development. And finally, CAP2, an actin-binding protein that regulates cytoskeleton dynamics, plays a role in cell proliferation and tuberogenesis. So its downregulation can be contributing to granulosa cell survival through anti-apoptotic mechanisms. And so the genes of this group all have to do with extracellular modeling and cell-to-cell signaling. And so, like they're the builders of the follicle, their differentially expression is showing us how these polycystic ovaries are not functioning correctly. And so what we learned today is that there is a very intricate molecular landscape of PCOS with critical roles of inflammation, immune dysregulation, and ovarian dysfunction. And this disrupted follicular growth is essential to the disease pathophysiology. With our immune gene group, may be contributing to systemic immune response and activation and ovarian dysfunction, reinforcing the importance of immune modulating pathways in PCOS, while the dysregulated follicular group may be responsible for the disruption of ovarian homeostasis, leading to anovulation and infertility. Identifying these differentially expressed genes in PCOS further enhances our understanding of the overlap between inflammatory pathways and follicular dysgenesis, suggesting a complex interplay between immune response and follicular maturation. And so the significance and possible clinical implications of these findings warrant further investigation, and future research is needed to explore these molecular mechanisms in genes in greater depth as potential biomarkers and to develop more precise diagnostic markers and targeted interventions to help in the disease management in PCOS patients. So these are my references, and thank you for listening. I'll take questions.